on with our general embryology session. This is part 3, chapter 3 from Langman Medical Embryology, and we have first week of development, ovulation to implantation. If this video is helpful, please like the video and subscribe. If you have any questions, you can post them in the comment. Thank you. Let's get started. First, we talk about the ovarian cycle. So to remember the phases of ovarian cycle, we have to remember the word follicle. And we write the word follicle like this. So first word we have F, and from F we have follicular phase, and it is at the start and it is under the effect of FSH hormone. Next is O, and it is ovulation, and we have L underneath it. So ovulation, there is LH surge, and this is in the mid of the cycle. At the end we have L, that is luteal phase, and that is under the effect of progesterone. Follicular phase is also affected by estrogen. First we talk about follicular phase, which is under the effect of FSH and LH from anterior pituitary, which is released from GnRH hormone, gonadotropin releasing hormone from hypothalamus, and they control and stimulate ovarian cycle. So at start of each cycle, there are 15 to 20 primary follicles that grow under the effect of FSH that affects the granulosa cell. FSH is not necessary for primordial to primary follicle, but without it, primary follicle may become atretic and die. To check the full details, you can watch the video above, in which I have discussed in detail about the primordial to primary follicle formation, then Graafian follicle. Out of 15 to 20 follicles, one follicle reaches full maturity and one oocyte is discharged. Others degenerate and become atretic. They are called corpus atreticum. So here is the one follicle that reaches the maturity. FSH also affects the theca interna, that is granulosa cell, and it secretes steroids, and these are estrogen, testosterone, and estradiol. These are theca interna cells in the inner layer. Along with ovarian cycle, there is also uterine cycle, and uterine cycle also has follicular proliferative phase, and in this there is thinning of cervical mucus that allows the passage of sperm, and there is LH release. And in follicular development, there is formation of Graafian follicle. In the mid-cycle, there is LH surge, oocyte complete meiosis 1 and initiate meiosis 2. Here is the primary oocyte which completes meiosis 1, and here is the secondary oocyte which initiates the meiosis 2, and there is ovulation, and here is the ovulation. You can check the full video to check the detail on how this cycle is going on. Next, we again talk about the step of ovulation. During ovulation, some women feel slight pain, which is called Mittelschmerz or middle pain. In the mid-cycle in ovulation, there is rise in basal temperature. Gonadotropin, FSH, and LH play a crucial role in ovulation. FSH and LH vesicular follicle grows rapidly to diameter of 25 mm to become Graafian follicle, that is mature follicle, and as vesicular follicle grows, it becomes the mature vesicular follicle or Graafian follicle. Primary oocyte completes meiosis 1. Secondary oocyte is arrested in meiosis 2 in metaphase 2 for the 3 hours. LH increases the collagenase activity, which causes the collagen digestion. Prostaglandin level increases, and there is local muscular contraction of the ovarian wall. Both of 1 and 2 steps in the mature vesicular follicle leads to the ovulation, that is release of the ovum. The cumulus ovorus that is surrounding granulosa cell around the secondary oocyte rearrange themselves and form corona radiata, which have projections like this. There is also stigma formation, which is surface of ovary bulge locally at the apex. Stigma appears, and the stigma is like this. Here is the stigma, and this helps in the process of ovulation. Secondary oocyte is in the meiosis 2. Next is the luteal phase. After ovulation, granulosa cells and theca interna cells of the ruptured follicle under the effect of LH hormone forms luteal cell, which contains yellow pigment, and they are called corpus luteum, and they secrete the progesterone and estrogen. Progesterone is very important here. Mainly after the follicular phase or proliferative phase of the uterine cycle, there is secretory phase that is under the effect of progesterone mainly, and secretory phase is important for the implantation of embryo. Next we have is oocyte transport. Before ovulation, fimbriae of the uterine tube sweep over the surface of ovary and contracts rhythmically. Oocyte transports from ovary to uterine tube. Cilia motion of epithelial cells. There is the cilia in the epithelial cells of the uterine tube, and the fimbriae contraction leads to the oocyte transport. Here are the fimbriae of the uterine tube that sweep over the surface of ovary. And here are the epithelial cells that contain cilia of the uterine tube and helps in the movement of oocyte. In the ampullary part of the uterine tube, fertilization occurs and the zygote moves to uterus in 3 to 4 days. Here are the ovaries. Here is the fimbriae. Here is the uterine tube or fallopian tube or oviduct. Here is the uterus. There can be two fates of corpus luteum. If the oocyte does not fertilize, no fertilization, there is formation of corpus albicans. Corpus luteum shrinks in 9 days after ovulation. There is luteolysis and forms corpus albicans, which is fibrotic scar tissue. Corpus luteum degeneration leads to decreased progesterone that maintains the secretory phase and causes the menstrual bleed. If there is fertilization of the oocyte, HCG, human chorionic gonadotropin hormone released by developing embryo that prevents the degeneration of corpus luteum. Corpus luteum keeps releasing progesterone until 4 months of pregnancy. Corpus luteum in pregnancy is corpus luteum gravidatus. Corpus luteum regress as placenta secretion after 4 months are adequate to sustain pregnancy. This one is important here. Next, the fertilization. It occurs in the ampullary region of the uterine tube. Ampulla is the widest part of the uterine tube. Only 1% of the sperm that is deposited in vagina reaches cervix. Here, the sperm is deposited and only 1% reaches cervix. It has to move to this part, that is ampulla of the uterine tube, to fertilize the egg. Movement of sperm from cervix to uterine tube occurs mainly by the contraction of muscular uterus. Some of the movement is performed by the propulsion of the sperm. Trip of cervix to oviduct or uterine tube can last for 30 minutes to 6 days. Here is the uterine tube and here is the uterus. After reaching the isthmus, sperm become less motile and they cease the migration. Here is the part isthmus, that is the contraction in the oviduct, and it is in the start. At ovulation, sperm becomes motile again due to substances released by cumulus ophorus of the egg. They reach the ampulla where the fertilization occurs. Here is the cumulus ophorus, the granulosa cells surrounding the secondary oocyte. Spermatozoa must undergo two steps, capacitation and acrosome reaction, to fertilize. So in capacitation, here we have a cap with digit 7. So conditioning of the sperm is called capacitation. It usually takes up to 7 hours. It occurs due to uterine tube mucosal surface reactions with epithelial surface of the sperm. Glycoprotein layer overlying the acrosome is removed. Here is the sperm with acrosome and here is the glycoprotein layer. In the capacitation, this glycoprotein layer is removed. Next, we have acrosome reaction. This is binding of zona pellucida to the acrosome. Zona pellucida is basically the glycoprotein shell around the cell. Cumulus ophorus or corona radiata is the granulosa cell around the secondary oocyte. There are three phases of the acrosome reaction. First is penetration of corona radiata. This is the corona radiata. Then is the penetration of the zona pellucida. This one inner layer is zona pellucida. Then is fusion of oocyte and sperm membranes. Here is the oocyte and sperm membranes where they fuse. So to remember the phases of acrosome reaction, we have the word acrosome. So for C, we have corona radiata penetration, which is the first step. For ZO, we have zona pellucida penetration, which is the second step. For ME, we have meetup of gametes or oocyte and sperm membrane fusion. Phases of acrosome reaction are very important and the sequence. So phase 1 is corona radiata penetration. In the start, 200 to 300 million spermatozoa deposited in genital tract. Only about 500 reach the site of fertilization and only one sperm fertilizes. Phase 2 is zona pellucida penetration. Release of acrosin enzyme from the acrosome causes zona pellucida penetration. Permeability of zona pellucida changes when sperm head contacts the oocyte surface by releasing the lysosomal enzyme from the oocyte granules, that is zona reaction, which we will discuss later. This prevents the multiple sperms to fertilize an egg, that is polyspermy. Phase 3 is fusion of oocyte and sperm cell membranes, that is meetup of gametes. Acrosome disappears in the acrosomal reaction here. Head and tail of the spermatozoa enter while plasma membrane is left on the oocyte surface. This is plasma membrane and this is head and the tail of the spermatozoa in the oocyte surface. Egg responds to three ways as spermatozoa enters the oocyte. First is cortical and zona reaction, which we talked earlier. Release of cortical and oocyte granules with lysosomal enzyme leads to oocyte membrane impenetrable to the spermatozoa, and there is impermeability of zona pellucida to sperms. There is a resumption of second meiotic division. Oocyte finishes second meiotic division with the production of second polar body and definitive oocyte. Here is the secondary oocyte here, and there is the definitive oocyte with the second polar body. Here are the other two polar bodies produced from the first polar body that is formed from the first meiosis. Its chromosome is 22 plus X. They arrange themselves in vesicular nucleus known as female pronucleus, that is 1N haploid. Here is the female pronucleus. Next is metabolic activation of egg. Activating factor is carried by sperm that leads to early embryogenesis. Spermatozoon forms male pronucleus that is also one-end and haploid. Male and female pronucleus DNA replicates and fuses and forms zygote that is two-end diploid. Here is the fertilization that leads to zygote formation. As sister chromatid moves to opposite poles, furrow appears. There is two-cell stage. Here the sister chromatids and the furrow appears and there is two-cell stage. The fertilization results basically in restoration of diploid number of chromosomes, that is 46 or 2n. There is determination of sex of the new individual. It is XX, it is female embryo. If it is XY, there is male embryo. Initiation of cleavage without fertilization, oocyte degenerates after the 24 hours. Next, we talk about contraceptive methods. There are barrier methods that can be condom, hormonal method that can be oral contraceptive pills. Intrauterine device can be copper IUCD and progestin device. Emergency contraceptive pill is 120 hours after sexual intercourse to prevent the birth. Sterilization is male vasectomy. We will talk them in detail later. Next is infertility. In infertility, we can use in vitro fertilization to treat infertility. Intracytoplasmic sperm injection is used to treat male infertility. Oligozoospermia is few live sperms. Azoospermia is no live sperms. Thank you.